So today we'll discuss control some questions on previous year CSIR-NET syllabus, that is control of gene expression in Operon concept. So first question: E. coli was grown in three different experimental conditions. In one, it was grown in a medium containing glucose as a sole carbon source. Second medium containing both glucose and galactose, and third was infected with fuzz. Actually, bacteriophage infect bacteria and destroy the bacteria. So bacterial growth will be decline. So you can see that graph should be for bacteriophage should be C. Now coming to that, back in E. coli actually use glucose as the major carbon source. If glucose is present in the medium, so it will grow exponentially, right? And it's have a, normally we see that bacteria have a three phase of growth: phase, lag phase, log phase or exponential phase, and stationary phase. Now if it is a medium, second is glucose and galactose. If the two source is there, now there should be some lag phase should be there. So this B curve should be for option for glucose as a carbon source, and second medium containing glucose and galactose. Glucose and galactose will be option A. So answer will be A is for glucose and galactose, and B is only glucose, and C is infected with fuzz. So answer will be option 2. Now question number 2. Now when you have created a fusion between the tryptophan operon, which encodes the enzyme for tryptophan biosynthesis, under the regulatory controls of the lac operator, right? Under which of the following condition will tryptophan synthase be induced in the strain that carries the chimeric operator fuse operon? Now understand the question. Here first is that there is a fusion between the tryptophan operon. Fusion between the tryptophan Operon encodes the enzyme for tryptophan biosynthesis under the regulatory controls of the lac operator. So here there is a fusion created under the lac operator, or you can see that in that way. So it's a, suppose these are condition. Now this one, this one. Now these are actually lac I gene. Lac I produce repressor, lac I. Now these are operator and promoter side. Now the under that lac operator and lac promoter, now all the TRP genes are TRP E, TRP D, all genes are under that control. Now here the control will be instead of, now here the control will be on the basis of lactose. Actually, as the regulator site is lactose, so when lacoperon actually on in presence of lactose, absence of lactose, the lacoperon is off, right? So lactose is during absence of lactose. If glucose is present, so gene is lacoperon is off condition, and so lacoperon and the tryptophan operon genes here, that's a chimeric tryptophan D, E, D, B. So all the other tryptophan synthetic genes will be on only when the presence lactose is present in the medium and glucose is absent in the medium, right? So answer will be option D. Next question. Now in a culture medium contain two carbon source. One is preferred carbon source, that is glucose. If bacteria use glucose, so do not require to synthesize all the necessary genes like synthesis of unnecessary proteins like expression of the operon, right? So it like G. LAC-Z produce beta-galactosidase enzyme. LAC-Y produce beta-galactosidase permease, and LAC-A beta-galactosidase trans-acetylase enzyme. But when it is non-peppered shifted into a second carbon source, so when a medium, if it is shifted into the second carbon source, it will, suppose these are bacteria growing, and now you are changing the medium. It's for first red graph for glucose. Now if you change the medium, some period of required for bacteria, that's lag phase is required to adjust with the environment. Changing sugar molecules, it's required some adjustment to the liquid, that is called lag phase. As a source, it will be answer C. A lag phase will be observed between the two exponential phase. Now next question. Now it's a in a strain of E. coli, a fusion between the lac and tryptophan operon took place in the and the new locus structure is shown below. The strain lack the wild type tryptophan operon. So same as now there is a fusion between, like previously we have seen operator and I is in lac, and the all other structural genes are changed. Instead here is tryptophan operon genes are placed. Now it will only synthesize in presence of lactose in the medium. And so tryptophan will be synthesized in the presence of lactose in the medium. So option A is correct. And tryptophan synthesis will be replaced in with the medium containing glucose. Yes, if it is a glucose is present in the medium, so lac operon gene is off condition. Same will be here. So answer will be option A and B. Now, next question. Question number 5. The following graph represent the expression of tryptophan synthase in E. coli cells in the absence and presence of tryptophan in the medium. Now, tryptophan synthase uh, is actually tryptophan operon under negative regulation, that is repressible operon. And a repressible operon, actually when tryptophan absent in the medium, then only tryptophan synthetic genes are on, right? So there is a two type of regulation is there. One is normal regulation, normal repression. Another is that the attenuation mechanism. Now there is a leader sequence in the tryptophan codon. The leader sequence is replaced, generally leader sequence have one important part in the leader sequence is the two tryptophan amino acids is there. If a two tryptophan amino acid is replaced with a alanine codon, suppose these are here the leader sequence, here these have a part of some TRP2 TRP codons are there, and these TRP codon are mutated to alanine codon, right? Now, in case of what will be the expression of tryptophan synthesis in the graph. Now, here you can see that there is a tryptophan, generally when the tryptophan level is low, there is a formation of hairpin structure, right? Two and three pairs, and then they synthesize the tryptophan synthetic gene. If it is a tryptophan is high level, there is a pairing of 3 and 4, and the transcription termination occurs, right? So in case of, if now here the TRP codon is replaces now by alanine codon. TRP codon is replaced to alanine codon, so there will be regulation will be lost. Now here the expression of graph level. So tryptophan, as the tryptophan is not there, so the graph should be option B will be the correct answer. But their expression level should be not too that much amount for in the absence of in cell growing in the absence of tryptophan and the presence of tryptophan. So this graph should be more or less that much amount. So answer will be here option B.